morning. This is Dr. Jingbo Louise Liu from Texas A&M University, Kingsville. So today we are going to discuss experiment number 9, the vitamin C analysis. The objectives are twofold. So our students will determine the amount of vitamin C in a vitamin tablet or a solution. So a fresh fruit and fresh vegetable samples. The second one, our students will understand the vitamin C for human health care. Vitamin C, like this graph showed you, this is the diagram of your vitamin C. The vitamin C involved in 2 electrons. So the movement of electron pairs in your deprotonation of vitamin C. So from this vitamin C, you are going to lose 2 electrons, so to produce a compound. So this vitamin C, commonly known as a reducing agent, is very critical for human beings. So the human body does not synthesize vitamin C. Therefore, we must obtain vitamin C from our daily life, from our diet. So benefits of vitamin C is called as the benefit of the vitamin C is also resulted from something called ascorbic acid. Your vitamin C is known as ascorbic acid, has a formula C6H8O6. This vitamin C is also known as diprotic weak acid. The benefits for vitamin C involved in several perspectives. For example, assists growth and development of teeth, gums, bones, skin, and blood vessels. Provides antioxidant, which can help to protect against cancer, heart disease, and stresses, and also help our human body to absorb ions and folic acid. There is more beneficial to consume sources of vitamin C, so the consumption is supposed to be consume vitamin C in a raw format. Cooked vitamin C can result in the leach out of your useful active chemical component. So some sources of vitamin C listed below. So usually in the beets, carrots, eggs, meats, milk, you do have a vitamin C. So 10 milligrams per 100 grams. So in some other like vegetables, so you do have 10 to 25 milligrams per 100 grams. Your sprouts, citrus, tomato, spinach have large amount of vitamin C. And also your chili pepper, sweet pepper. So you have your kiwi, so they have a different amount of vitamin C. So they listed the sources of your vitamin C which you may uptake in your daily life. Again, your vitamin C, also called ascorbic acid, the formula is C6H8O6. This is the structural formula for your vitamin C. By nature, vitamin C is colorless, is water soluble, and this chemically speaking, vitamin C is a reducing agent. Vitamin C itself is going to be oxidized just in air by your oxygen. If you increase temperature to more than 190 degrees, between 190 and 192 Celsius, your ascorbic acid, vitamin C, may decompose. So again, cooked food may result in the leach out of your vitamin C. So I want to also recall several fundamental premises, several fundamental terminologies during your titration. Analyte, titrant, primary standard, standard solution, and hygroscopic. So analyte is the substance which you are going to study, the chemical component which is going to undergo analysis and to be measured in your analytical procedure. Your titrant is also known as standard. This one you know is concentration. So the volume of the standard solution is used to determine to react with the measured quantity of your unknown sample analyte, and adding the titrant into your calibrated burette to determine the exact amount of your standard solution. Primary standard refers to a chemical, a substance which has very high degree of purity, relatively large amount of molar mass, usually should not be a hygroscopic, so and also can react with your unknown sample in a predictable way. Standard solution is a measured number of moles of your substance which is present in a measured volume of your solution. The concentration is known. Standard solution usually reacts with your substance, the analyte, to be analyzed. Hygroscopic related to the absorption of water. Some compounds, if you leave it in the atmosphere, you figure it out, it become crystallized, or sometimes they stay together, adhered together. So this is due to the absorption of water from the atmosphere. This phenomenon is called hygroscopy. About our research, our experiment in this study, you related to four key steps. So number one, you are going to prepare a standard solution, potassium iodide, and you add excess iodide into this iodide in the acidic solution. You are going to produce iodine, I2, and water. This is your first important equation actually. And your sample of your ascorbic acid is going to be treated with the known amount of the solution. The ascorbic acid usually can be reduced by itself, is going to be oxidized and reduced some other party into the negatively charged ions. For example, your ascorbic acid can reduce this iodine into the iodide. By itself becomes C6H8O7. And this the reaction is 1 to 1 to 2 in terms of your iodide. So again, be careful with your balanced reaction. The first balanced reaction, 1 mole of iodide to 5 moles of 1 mole of iodate to 5 moles of iodide, 3 moles of iodine, I2. And your ascorbic acid again is acting as a reducing agent. And your I2, iodine, somebody also called iodine. So this is oxidizing agent. This is another important reaction for you. So the remainder, usually this I2 is in excess. You are going to study the back titration. I2, the remainder I2, you are going to be titrated, use a standard solution, sodium thiosulfate. So and then you produce another colorless I negative. Actually, this I negative should be I3 negative, triiodide. That's what you learned from previous study. And you produce triiodide simplified as iodide and S4O6 negative. This is another important equation. So be careful, this is 1 mole to 2 moles. The amount of your ascorbic acid equals to the generated iodine, this total iodine, minus the excess iodine. That's how you are going to determine amount of your ascorbic acid. In terms of our procedures, I want to particularly mention that you are going to use potassium iodide. Why you use this potassium iodide as your primary standard, and you have to do the back titration because this solution is highly pure. You have a very relatively large relatively large molar mass. This compound is not hygroscopic. This compound can react with your iodine I2 in a predictable way. And then, in a predictable way. Sorry for that. And then your sodium cell sulfate solution also need to be prepared and standardized. If we give you the tablet of your vitamin C, make sure you measure this tablet carefully and also ground it into very fine powders. And then you are going to do 2 different titrations. So that's essentially what you are going to do in the lab. About some examples for the calculation I listed in this slide. So first of all, you assume you have 6 ounces of vegetable juice, which can provide 35% of recommended RDA of your vitamin C. I want to determine how many milliliters of this vegetable juice will provide 100% recommended vitamin C. RDA meaning recommended daily allowance usually should be more than 60 milligrams. So there are also a couple different units you need to know about the conversion. 1 ounce is approximately about 29.57 milliliters. So you are going to convert, we gave you 6 ounces, you convert to milliliters, and from milliliters, total milliliters, you convert to the vitamin C milliliters to the total total solution volume. So you use that 177.42 divided by your 35%, because your total juice provided 35. This 506.91 is the total amount of your vegetable juice. And then we are going to continue to working on this. So I have showed you several different steps. If you have 4.92 grams of lemon juice, so you are going to be added into the solution composed of 50 milliliters of potassium iodate. So this potassium iodate primary standard has 0.0102 moles per liter. And also you have excess potassium iodide. This potassium iodide total amount can be determined by the calculation. And then the titrated, you are going to using these two to react to produce I2. The I2 will be titrated by using the sodium sulfate. So in that case, you can calculate the number of I2 in excess. And from there, you calculate your vitamin C amount. Let me go to the example to do the calculation. So again, you have 1 mole iodate, so produce 3 moles of iodine. So 1 mole vitamin C reacted with 1 mole of iodine. So this is generated, so they fully reacted. And then this amount is unknown. You use the total generated ones minus this excess one. And then you correlate, this is 1 to 1. This number of iodine essentially is the same as the moles of your ascorbic acid, vitamin C. So here is our calculation. First of all, you are going to determine the moles of your potassium iodate. We gave you the molar concentration. You did the reaction, which is 25 milliliters. Therefore, you determine the moles. From this balanced reaction, you can determine the total moles of generated iodine. So which is 1 to 3. This number you multiply by 3. And then you calculate the moles of iodine, which is titrated with your sodium sulfate. Sodium sulfate is again a standard solution. You know the molar concentration. the amount in milliliters is the one you did the titration, and then you determine the moles of your I2 excess. The I2 excess, this is a 1 to 2, therefore this moles of your Na2S2O3 will be 2 times of your iodine. So you calculate the excess iodine and then calculate the difference. This difference is the same as the, let me go back, is the same as this iodine. The total, the generated minus the excess gave you the iodine moles. This molecule iodine reacted with your ascorbic acid in 1 to 1 molar ratio. And then you determine the moles of your ascorbic acid. So we did it, we determined the moles. So here is the moles of your ascorbic acid. As we mentioned, the moles of iodine is the same as your ascorbic acid. So from these moles, you can determine the mass, you can determine the percentage. The moles multiplied by the molar mass of your ascorbic acid gave you the grams. So you use the total grams of your lemon juice or whatever we gave to you. So in our case, we gave you the lemon juice, which is 0.246, and the grams of your ascorbic acid you calculated. Use ascorbic acid mass divided by lemon juice mass multiplied by 100%. So you produce, you created the percentage. That's how you are going to determine the RDA from your calculation. So again, I want to mention there's 4 major chemical reactions. 3 major chemical reactions. So one important calculation that should be very important for our students to determine the vitamin C. Thank you so much. That's all for today. Stay safe